Okay, so good afternoon. I just want to mention that the final exam of this course, which I've been touting for the past couple of weeks, is actually electronically on the website. You just Google Demystifying Medicine or go to the listserv. And if you've been to at least 50% of the sessions and you pass the exam, which is not going to stress you unduly, and maybe you'll learn something in the process, and you can take it over and over again, you'll get a certificate saying that you've been here, and we're all grateful for you. So next week is really the last session, and we've collected a bunch of questions that some of you submitted about things that you would like to hear from the speakers. Now, the speakers are an extraordinary group. It's the Institute Director of NINDS, the Institute Director of the National Institute of Mental Health, John Gallen, who heads the Clinical Center, and Bob Balaban from the National Heart and Lung Institute. Now, the whole point of it is that this will be sort of a freewheeling discussion, because what you want people have asked them to talk about are what are the opportunities as they see it in their fields, where are the exciting, expanding areas which should be of interest to you all, and what opportunities may exist for PhD scientists in particular. And they're very realistic people, so they'll talk in terms of what the problems are today as well. At any rate, it's up to whoever comes, and usually we've had a very good crowd of very active people. Last thing, if anybody has any comments, suggestions, criticisms, good, bad, indifferent, about the course itself, we'd all appreciate hearing from you, particularly suggestions of topics and people whom you might like to have participate next year. Okay? So this is the last time. No, next week you'll see it again. The Brooklyn Bridge, which is sort of the symbol of what we're trying to do to link cutting-edge opportunities, excitement, and scientific development, and major health problems. Sometimes it's the science that drives the clinical. And today, as I see it, it's sort of an extraordinary coming together, a coming together of exciting advances in understanding what the mitochondrion does, other than make ATP and control energy metabolism, but the biology of the mitochondrion. And for those of you who are used to seeing pictures in a textbook way back, those mitochondria weren't moving, they were just sitting there. And it turns out, of course, in biology, everything is moving. Our second speaker today, Jennifer Lippincott-Schwartz, I think one of the main themes of Jennifer's really exciting studies in many areas of cell biology is the dynamic nature of the processes going on within a cell, and the fact that you have your hands tied if you are looking at these only from one dimension. So her laboratory blends biochemistry, live cell imaging using an incredible array of technologies, some of which were developed in her laboratory, biophysics, and increasingly a somewhat pathophysiologic orientation to what the problems and questions are. And then, on the other hand of the bridge, for those who don't know, by the way, this is the Brooklyn Bridge, the most famous bridge in the world. On the other side of the bridge, in medicine, this is a beautiful example of the fact that there have been innumerable descriptions, usually in the pediatric or young adult literature, of patients who have a myriad of symptoms, not always, but usually focused on impairments of metabolism, of energy. But it may involve neurological features, sometimes solid organ features, energy, growth, a whole array. And the literature through many, many years is filled with these. And they have people's names attached to them because they're usually the clinicians who describe them. And it's precisely the accuracy of those descriptions that has now made it possible with the tools of genomics to begin to ask questions at a fundamental level of what are these diseases and what are the gene mutations coding for in the way of a protein, and what does that protein do in normal metabolism, whether it's in the brain or elsewhere. So the clinical descriptions in the area of what we call mitochondrial diseases, sort of primary mitochondrial diseases, because ultimately every disease knocks off the mitochondrion, and probably that's why we die. But in those primary diseases, it's the clinical description that form the basis. Otherwise, one would be at a loss just screening people for mutations but not having a clue as to what the syndrome or the symptom is. So this is a very good unification, okay? So, where is... All right, I want to tell you a very quick story to illustrate this. This is Monument Valley in Arizona, the home of the Western Navajo. As the story here, an American story of a poisoned land and people betrayed. That's a long story we're not going to get into today, but it has to do with things like open pit uranium mining, lack of support, malnutrition, all kinds of things. But this is a highly inbred population. And in the next slide, thank you, it's almost 20 years ago, colleagues and I were visiting there because of youngsters like this who were incapacitated by a combination of neurological and metabolic and mainly liver diseases. That's what attracted me. Can we have the next one? And so we studied some of these, and here's some of the families involved. Particularly family 2 was the most accurate, but it was very hard to get a family history from these people, but most of the folks affected were quite young. And so this was a combination of neurologic problems and liver problems. There's a lot of liver disease in this reservation, alcohol, hepatitis, very hard to dissect it all out. But the patients had impaired growth, motor and sensory neuropathy. That meant they couldn't walk, and they had sometimes no pain senses in their body, often cognitive impairments, muscle atrophy, which was due to the neurologic problem, and due to loss of sensation in the cornea, frequently they couldn't see because they had scarring there. And then they had this fatty liver, which progressed to cirrhosis even at the age of 15, 16 years old. Very, very strange. And a metabolic abnormality which tended to lead to hypoglycemia and acidosis, particularly if somebody had an infection. And it was not associated with great longevity. So we and a group of others, next slide, studied this thing extensively in the pre-genomic era. We did all kinds of metabolic studies, came to the conclusion that there was something wrong in some of the transporters that were present. Well, it turns out that later, in 2006, this is the group at Columbia that has done a great deal of work on genomics of mitochondrial primary diseases, and they describe that this is caused by a mutation in MPV17 gene, which has something to do with mitochondrial DNA maintenance. And if mitochondrial DNA is having problems, there are going to be all sorts of, you might say, downstream effects, like an oxidative phosphorylation. And in those days, we were looking way down the end of the track. So this is an example of an incredible clinical description by very astute pediatricians in a very difficult area where it's difficult to get accurate information, who got enough information that permitted some family studies, and then to recruit people to do what in those days was the state-of-the-art sort of metabolic studies, but ultimately led to the identification not just of this disease, but as we'll hear more about it later. And of course, the next thing is, what does this gene do? And that's still a mystery. So, next slide. Well, this is all familiar to you, so I don't have to go over what the mitochondrion does to make ATP, but that's going to crop up again in the discussion. And this is really to introduce Jennifer also, because so with this business of movement, and something that now is widely accepted in cell biology, but not so long ago, particularly prior to the discovery of green fluorescent protein, which Jennifer was among the first to exploit to answer dynamic questions, and then it expanded into many other kinds of probes that could be examined in living cells. So the theme of the whole thing, at least I think, of her research, is somewhat correctly stated by Galileo, who said, and yet it moves. Of course, he was talking about the moon, but nevertheless, within the cell, everything moves. And Heraclitus, long before, said everything is in flow, nothing remains the same. These are great ideas, but it took hundreds of years to put them into the reality of something you could see. So we're very grateful to have both of you here, and we'd appreciate, maybe you want to start?
Sure.
Yeah. Oh, wait a minute, I forgot to say.
I can introduce myself.
That's right. I mean, Lynn Wolf is the first speaker. Lynn is a nurse practitioner who works at the NIH in a remarkable program that you should all know about called the Undiagnosed Disease Program, and she'll tell more about it.
So hi, everyone. So yes, I do work in the Undiagnosed Diseases Program, which we just finished our pilot study of 5 years, the brainchild of Dr. Bill Gall. It ended September 30th of 2012, and now we've sort of entered a new era, which I won't discuss. But this is Paula and Liz. Liz happens to be a patient that we saw in the Undiagnosed Diseases Program, actually pretty close to its inception, about 3 years ago, I think, right? And she has a mitochondrial deletion syndrome called Kearns-Sayre syndrome. I've not been accused of being quiet usually. So what I'm going to do is I'm going to first just ask some questions from Liz and her mom about their experience being diagnosed with a mitochondrial disease and living with a mitochondrial disease, and then I'll talk to you about some of the clinical things that we know about and are doing to try and diagnose other mitochondrial diseases on the clinical side. Paula, I'll start with you, but then Liz, I want you to jump in. Tell us, when did you first notice any health problems with Liz? You guys can just pass it.
Okay. When Elizabeth had her adenoids out, she was 3 years old. And when she came out of surgery after having anesthesia, she didn't come out of it too well at all. She was, we brought her home, and she was kind of like a rag doll. The next day they readmitted her because she had no muscle tone in her body. And it seemed to be that after that episode, her eating stopped. You know, she started having difficulty eating. By the age of 5, she started to have like some ptosis with her eyes, and we started going.
What does that mean, ptosis?
Ptosis with the eyes means, you know, she had really big blue eyes, big round eyes.
Show them your eyes.
Right, so her eyes started to droop a little bit, and they thought it was a neurological problem. Then they thought maybe myasthenia gravis. These are all just different diseases that they were sending us to different hospitals checking. We ended up at Yale New Haven for, I think, 6 years. And after having numerous amounts of tests, they came up with that she had an eating disorder and put a GI tube in her. And then we ended up, after that, going to Columbia, because, you know, we're in the Northeast, so we have a lot of good hospitals around us. And at Columbia, we actually went into the Mito clinic twice, and they missed her diagnosis. And at that point, our pediatrician sent a letter here to NIH, to the Undiagnosed Disease Program, and we sent in photos from birth till present at that time, and you could see the progression with the eyes and the weight and everything that happened with her. And after being here for 1 week, Liz was diagnosed with Kearns-Sayre syndrome.
Liz, do you want to add anything about your diagnosis for everybody? You're going to be quiet? Okay.
Do you want to talk about how it is for you?
How is it now? Tell us, how is it now that you know your diagnosis?
It's kind of hard for me because I can't really do that many activities with my friends and do a lot of things.
Okay. Do you want to ask anything else? Anybody else want to know anything? I'm going to talk a little bit more about Kearns-Sayre's, and I can say a couple of things about Liz, you know, right now, just so that you know this, and then you can pop in. So, so Liz was being followed by an eye doctor, and so she had not only progressive lid ptosis, but she also had some changes in the back of her eye. So her optic nerve has gotten more atrophied, meaning it's wasted away. Yeah.
Could you talk to the mic?
I've got this one on, so I don't need it. Yeah. If I do it, it's going to squeak, so I won't do both. She also has some retinitis pigmentosa, and I have a good picture of that, so I will show you that, and that impacts her night vision. And so she had eye things happening, and the eye doctors were monitoring her, and she had growth things happening and was being followed basically by an endocrine service. And the interesting thing here, which is important for you all to know, is that she didn't see a neurologist or a geneticist actually until she went to Columbia twice and then came here and saw a number of different neurologists and geneticists. But the person that referred her to the Undiagnosed Diseases Program was her pediatrician, who was the one person coordinating her care, who said, all these eye things are happening, all these growth things are happening, and why is this not one thing? Why can't this be one syndrome? And that was how she got referred to us. When her mom talks about the GI tube, she has a gastronomy tube, which means that she actually has a tube that goes directly into her stomach, and she needs food, liquid food, overnight, every night, to maintain her health and her weight, because she just can't eat or drink enough during the day because of the energy that she uses. So Liz, I'm going to ask you one question that I know has been an issue. So how do you feel when your friends are having sleepovers, or they want you to go swimming and you're worried about a bathing suit or what your friends might see with your G-tube and needing tube feeding at night?
Most of my friends, they know about it, so they know that they can probably see through.
Okay. And has it ever bothered you?
No.
It did used to bother you.
It
Yeah.
bothered you.
Remember when you were younger, you didn't want people to see your tube?
Yeah.
We went through a little bit of a growth spell where we wanted to wear a bikini, but we were afraid to show off our G-tube. So now we don't do that. But there's other things that we've been able to do that have helped. Do you want to add anything else?
I just wanted to add that the everyday care for Liz, you know, when you have a mitochondrial patient, is you have to constantly be like one step ahead of her, because things change really quickly, like the heat intolerance, cold intolerance. You know, we could be going somewhere and all of a sudden, you know, she gets overheated, and it's not like you can just cool her right down. It gets very dangerous. Dehydration can happen within a couple hours. She can be okay, and then all of a sudden she can be severely dehydrated. So it's as a parent, it's a lot of work to, you know, we always travel with water, we travel with snacks, we travel with clothes to put on, clothes to take off, ice packs in the summer. You know, she has like a cooling vest to wear because she wants to be outside with the kids. And in order for her to be able to do that, you know, she has to have this cooling vest, which they've now made for mito patients. Life is not, you know, it's not like the other kids. She can't play sports in school. She's limited to, you know, how long she stays there. And a lot of times the school's willing to let her go and take naps if she needs to during the day. You know, so it's a different type of lifestyle for these kids.
So in fact, bringing that up, I'll just tell you this briefly and then I'll start my talk. So in order for us to keep Liz safe so that she could come and share her story with you, she actually was flown in on Sunday, so she had a full day of rest. She saw me in clinic yesterday morning early. She had some blood work done, and then she went back to the Children's Inn for rest until earlier today, and she did take an afternoon nap for about an hour at about 1 so that she would have enough energy to walk with me from building 10 to here and be able to share her story. So her energy is very limited and doled out carefully.
How old is she?
14.
The calorie intake,
So that's a really good question. So what we ended up doing for management of nutrition is not only addressing calories, but we addressed her protein load. So Kearns-Sayre syndrome can have a number of endocrinopathies associated with it. Some of the patients are hypothyroid, like Liz. Some of the patients develop parathyroid failure, which she hasn't done yet, but we've been monitoring for. Some of the patients develop diabetes, which we've been monitoring for, and she sort of is borderline. So we have her getting 4 cans of Glucerna diabetic adult formula that you see advertised on TV. It has about 11 grams per can, 11 grams of protein per can. So she gets about twice as much protein as a normal 14-year-old might get if they're not, you know, like an athlete. And she gets complex carbs that are very slow. And her total calorie and fluid intake is managed actually by our metabolic dietitian, and we go up and down. But Mom thought she was getting a little chunky and cut her from 4 cans to 3 cans, plus her grazing all day, and she went from 84 to 79 pounds in just a week. And so that 4th can really becomes quite critical.
Can you tell us what is the gene that's defective?
Yeah, so Kearns-Sayre is actually not a specific gene. I'm going to talk about it a little bit. So Kearns-Sayre is a deletion syndrome. So the mitochondria has its own DNA, which will actually be in my presentation, so maybe I should just do that, because it will make more sense if you see it. Okay, so I'm not sure how to turn this one off, so I'm going to, like, drop it. Or you want to have it, Lynn? Yeah. Okay. You can stay there, or you can go if you want. Okay.
Come watch the presentation.
Yeah, I was going to say, so I will also say that one of the things that we were able to do importantly for Liz and her family is the United Mitochondrial Disease Foundation is very, very active. And they have a 4-day meeting every year with an overlap of the basic scientists and clinical sessions with family sessions. And so Liz and her mom and her dad have actually been 2 years. So they have been very active and had the opportunity to meet with a lot of other mitochondrial physicians around the country and genetic counselors and nurses and dietitians. And they've met a lot of basic scientists that come to the meeting and talked a lot about mitochondrial disease management, not just with us here, but with other people around the country. All right, so let me start this talk. And I heard there was a thing, but I don't see it. So this is a stagnant, healthy mitochondria by electron microscopy. And I have some pictures of some abnormal ones, but mine aren't moving. So I don't know. Do you have movies of moving mitochondria? Okay. So I'm going to give you just some basic facts about mitochondrial disease in general, because we think of these as rare disorders, but they actually really are not. So a lot of people that work with mitochondrial disease around the world have been trying to get a good handle on how heavy a disease load this is. And in 2000, there was a publication that found worldwide we have a prevalence of about 11.5 per 100,000, which means 1 in 8,500 of us will have a mitochondrial disease at some point in our life. In 2006, the incidence was 1 in 5,000 of very specific changes in oxidative phosphorylation. So those would be patients that have documented electron transport chain defects. And in 2008, there was an interesting study done by Chinnery's group in England looking at carriers of mitochondrial DNA mutations. And what they did was they took the newborn screening cards from the entire United Kingdom and did mitochondrial DNA analysis, and they found that 1 in 200 persons, which translated to 1 in 10,000 of the adults in the United Kingdom, actually did carry a pathogenic mitochondrial DNA mutation. The question is, why are they not all manifesting disease? So we'll talk about that as this moves on a little bit. So in the United States, there's about 4 million children born each year, and 4,000 of those will develop mitochondrial disease. But we also have about 50 million adults that suffer with either a primary or a secondary mitochondrial disease, because as many of you probably know, we have mitochondrial failure in lots of things like cancer and infertility and insulin-dependent diabetes, heart diseases, blindness, deafness. The list is quite extensive. And as Lynn referred to, we do also believe that mitochondrial dysfunction ultimately is why we all age and die. So the functions of mitochondria that we all learn about from very early, I think, in high school is that you need your mitochondria as the powerhouse for your cells, and that's where your ATP comes from. But as it turns out, the mitochondria have numerous other functions that are really critical to growth and development. And so I've listed them all here. And so this explains why when we look at our patients in the clinic, we see lots of symptoms across all the body systems. So consequences of dysfunction include decreased ATP production, which, as you've heard from Liz and her mom, impact on how you can thermoregulate, how much activity you can have in a day, how your body can utilize its nutrients. And so what ends up happening is that decreased ATP production becomes critical to a lot of the symptoms and the changes in daily living for these patients. There's also an increase in reactive oxygen species, which is felt to contribute to ongoing mitochondrial damage and loss, which can potentially make mitochondrial disease worse. Lipid peroxidation is basically the breakdown of the fatty layers in our cell membranes, and so you have ongoing cell destruction. There's changes in the membrane potential, so the electrical capacity of the cell membrane, and apoptosis is programmed cell death. So when the mitochondria are not healthy, our cells just don't think they should be hanging around anymore, and so those cells die off, and then you have even less mitochondria. So here's the mitochondrial genome. And what I want to point out, although this one's kind of weak, is right here is the sporadic deletion that's associated with Kearns-Sayre syndrome, progressive external ophthalmoplegia, and Pearson's anemia, which are now believed to be spectrum of exactly the same disease. So it's just how much of the deletion do you have, how big's your deletion, and that manifests your symptoms. So this is the mitochondrial DNA. It's very small, but remember there are numerous mitochondrial DNA in every mitochondria that are in our cells, and there are numerous mitochondria inside all of our cells. So we have lots of copies of this particular mitochondrial DNA, and so some of the DNA will have the deletion, and some of the DNA will be what we call wild type, normal mitochondria. So the normal deletion in this DNA is somewhere between 2.5 to 5 megabases, and what ends up happening for Liz, she has 3.9 missing, which takes out ND4 this subunit and part of ATP6. So she slices through like that, and that explains her symptoms. So she affects complex 1 function, complex 4 function, and complex 5 function. Oops. The mitochondrial genetics is really complex, and I didn't put all of it in here, but we can discuss it a little bit. So mitochondria are inherited from our mothers. It's all in the eggs. There's very little in the sperm, and where it is in the sperm is the tail. So post-fertilization, the tail drops off, and we inherit most of our mitochondrial DNA from our mothers. There is also a unique genetic code to the mitochondrial DNA that's different than our nuclear DNA. There is something called replicative segregation, which I'm going to show you some pictures of, and some heteroplasmy as well. There's a higher mutation rate in the mitochondrial DNA than in the nuclear DNA, and in the mitochondrial DNA there are less repair mechanisms. So our nuclear DNA has lots of mechanisms to repair problems, but the mitochondrial DNA doesn't have as many, and so if there is damage, a mutation, that perpetuates. And then there seems to be a threshold expression for phenotype. So you have to have a certain balance of bad mitochondria to wild type or normal mitochondria before you have symptoms. And that varies disease to disease. I'll show you one example of a clinical syndrome that we do know how to look at the proportion of mutated DNA. So heteroplasmy is just the percent of abnormal mitochondrial DNA to the total mitochondrial DNA, and it can vary from cell to cell and also from tissue to tissue. And the percent of abnormal mitochondrial DNA increases over time in normal aging as somatic mitochondrial DNA mutations occur and just don't get repaired. The percent of heteroplasmy may indicate age of onset of symptoms. So we see patients with mitochondrial diseases that are very severe in the newborn, and then we can see patients being diagnosed with mitochondrial disease into their 60s and 70s. So this is replicative segregation. So unlike the nucleus, so if you remember the nucleus, the nuclear DNA will divide in half and you have equal amount of DNA in each sister cell. But because we have numerous mitochondria in every cell, and we have some that have disease and some that are normal, when the cell separates out, you get a different dispersion of disease versus wild type, and that perpetuates, and so you can't predict really well how the patient is going to represent. So mom can have a mix of diseased mitochondria and wild type and have no symptoms at all. And then as she divides her cells, and think about the eggs, then you can have some of those eggs or those cells develop different percentages of disease versus healthy mitochondria. And then depending on that, she can give to her offspring or even to a specific tissue. So you can have some mitochondrial diseases that only impact the eye or only impact muscle or only impact the GI tract. So even though this talks about the whole being, think about this as it could also be tissue specific. And so what ends up happening is post-fertilization, you can assume that the child that has more diseased mitochondria than wild type might have more severe disease, and a child that inherits more of the wild type or normal mitochondria will have less disease. All right, the mitochondrial electron transport chain is called complexes. I'm not going to spend a ton of time on this, but just to show you that these are multiple complexes that make up multiple proteins that make up each of these complexes. And so the function of each complex is not only dependent on how the unit works when it's together, but also how some of these units are assembled. And because some of them are actually regulated by mitochondrial DNA and some are by the nuclear DNA, you can have mutations in either your mitochondrial DNA or your nuclear DNA impact on the maintenance and assembly of the complexes or the function of the complex once it's assembled. So even if it's assembled correctly, it might not give you the energy that you want at the end of the day. So there's a lot of mitochondrial symptoms, and I've kind of listed these. And when we are seeing a patient in clinic, this is what we're looking for. So we're looking for constitutional symptoms, so general problems with growth, which you probably didn't notice, but Liz is actually well under the 3rd percentile for a 14-year-old, so she has short stature. Cachexia is really just failure to thrive. And so it's very unusual for anybody to need a G-tube and tube feeding at the age of 9. So Liz was diagnosed with failure to thrive at the age of 9 and had a G-tube put in before she was ever diagnosed with her mitochondrial disease. So in hindsight, we were able to put that together. Bone marrow failure we see in a lot of patients. Sideroblastic anemia, which is a very specific type of anemia that you can pick up on a peripheral smear, so the hematologist will see that on a slide. Lots of problems with the endocrine glands working. You can have lots of problems with the nervous system, including early strokes and migraine headaches, which Liz has had, and stroke-like episodes, which Liz has had. Lee's syndrome is actually a description of a brain degradation, neurodegeneration that was described and is seen in a lot of different types of mitochondrial disease. Seizures, early onset dementias, lots of different movement disorders, balance problems, hearing loss, and then eye problems, which we actually talked about with Liz. Ataxia is an unsteady gait, loss of reflexes, which we also heard when talking about. You can have problems with cardiomyopathy, which is an enlarged heart muscle, so it doesn't pump as normally as you would want it to pump. And you can have heart block or other arrhythmias, which I think we mentioned. Did we mention? So Liz, about a year after we made her diagnosis, developed heart block, which is also very common in Kearns-Sayre syndrome. And so she actually has a pacemaker in. Easy fatigue, we've talked about, exercise intolerance, weakness, low tone, muscle pain and cramps. Rhabdomyolysis is actual breakdown of your muscle, and you get an elevated chemical called CK or CPK that we measure in blood. That's not a typical symptom of Kearns-Sayre's, but you see it in other ones. There's lots of gastrointestinal problems, and there's some kidney problems as well that you can see. And not every patient with a mitochondrial disease will have every single one of these symptoms, but these are the things that if you're in clinic with a physician or a nurse practitioner that look at these types of patients, these are the things that are sort of red flags for us as we're talking to the families and the patients that make us think maybe this is mitochondrial disease. The problem is that there is incredible variability in presentation even in the same family. So you can have children in the same family that will have slightly different symptoms because of the things we've already talked about, because maybe they inherited a different load of wild type versus diseased mitochondria, or maybe for some reason their diseased mitochondria are primarily in muscle and somebody else is having more GI symptoms. So it's really hard, and we do need our genomics to help clarify some of this. Symptoms of mitochondrial disease are usually progressive, and there's no cure currently. So anybody out there wants to work for a cure for a mitochondrial disease, we'd be very happy. And then correlation with molecular defects is variable, and we'll use MPV-17. MPV-17's now been described for almost 10 years, and we still have no idea what that protein does. And there are lots of newer mitochondrial disorders where we've found genes that we know we can localize to some part of the mitochondria, but we don't know what the proteins do. So there's multiple types of mitochondrial disease. There's about 400 currently published clinical syndromes, and they fall into these general groups, but I'm not going to pretend to tell you that this is an inclusive list because it changes pretty quickly. But the older disorders are recognizable mitochondrial syndromes from mitochondrial DNA point mutations that were first starting to be described in the late 50s and early 60s, which Kearns-Sayre's was described in 1959. And so they have these very nice acronyms. So MELOS, MERF, NARP, LHON, MID, they sort of roll off your tongue, right? But what you need to know behind there is that MELOS is a myoclonic epilepsy with lactic acidosis and stroke-like syndromes. So aren't you glad you don't say that all the time? So, and MERF has red ragged fibers, and NARP is a neuropathy ataxia with retinitis pigmentosa, and so you learn these as you do them all the time. The mitochondrial DNA deletions and rearrangements include the Kern-Sayers, chronic progressive external ophthalmoplegia, Pearson's anemia, and then we have a number of mitochondrial diseases that are related to nuclear DNA. So we have an autosomal dominant optic atrophy. There's a specific type of methylglutaconic aciduria. There are codependent functions, so CoQ, which many of you guys probably see now. It's very common in hair lotions and face creams, but everybody forgets it's really critical to mitochondrial function and is a normal chemical in our bodies. Electron transport chain deficiencies can be single or multiple, and complex 1 is the most, isolated complex 1 is the most common across all ages, but frequently more than one electron transport chain complex is involved. Then we have a number of mitochondrial depletion syndromes, which is where the MPV17 gene sits. There's now numerous genes that are associated with the mitochondrial depletion syndromes. And so what happens there is, for whatever reason, those genes impact on either mitochondrial DNA maintenance or the mitochondrial DNA biogenesis. And so in our mitochondria, if your mitochondrial DNA is not being replaced or not maintained, those mitochondria die, and then you end up with a mitochondrial depletion syndrome, which are usually more severe than some of these other point mutations, because you literally run out of mitochondria in your cells. I worked with a neurologist once that told a family, imagine a chocolate chip cookie, all the chocolate chips are your mitochondria, and somebody just eats those chocolate chips, and then you just have the cookie dough. And then what can it do? It just crumbles. Okay, so then there's some carrier proteins, and there are multiple carrier proteins now. This acetyl-CoA synthase group is now probably close to 20 genes. We still don't know what all those proteins are doing. We have associated proteins with the mitochondria that cause mitochondrial diseases, and then we have secondary defects, like I talked about early, in neurodegenerative disorders like Parkinson's and Alzheimer's and cancer and cardiomyopathy. So there is a threshold effect, and now you're starting to see these are some abnormal mitochondria with paracrystalline inclusions, and these are just swollen, misshapen mitochondria. So some tissues require more energy than others, and so those are the symptoms that are going to show symptoms first. And so when you think about those, you can use Liz as our example. So the nervous system requires tons of energy, and the eye is part of the nervous system, and so is hearing. So you have lots of brain problems, lots of eye problems, lots of hearing problems. The peripheral nervous system, what gives you balance and your ability to walk, that also is high energy. So muscle, so your skeletal muscle, we have to stand with our skeletal muscle, we have to move with our skeletal muscle, so that is another high energy tissue. The heart muscle, and then remember that you also have smooth muscle in your GI tract and in your breathing. So some patients with mitochondrial disease have lots of breathing problems too, even though I didn't necessarily mention that. And then the endocrine glands that are very important to normal growth and development are also high energy, and so we see lots of developmental and growth issues. And then our kidneys, so and then liver and other things. But these are this is why you see the symptoms that you see, because these are the tissues that need the most energy to function properly. So in the NARP syndrome, we know in a certain mitochondrial DNA mutation, this T8993G, the clinical phenotype will vary by the mutant load, how much diseased mitochondria you have in comparison to your wild type. And so what you can see is if you have 0 to about 60%, and that can be variable in some patients, you will be considered normal. You won't manifest symptoms. But somewhere, if you carry 60 to 75% of this mitochondrial DNA mutation, you might have isolated retinitis pigmentosa, and that would be identified by your eye doctor once you start having night vision problems is the most common thing first. But if you have 75 to 90% of a mutant load of this particular mutation, you have a full-blown NARP syndrome, which is a late onset syndrome you usually see in your 20s to 30s. And then you might have, from 90 to 100%, you have a full-blown infantile Lee syndrome, which is usually fatal before the age of 2. So this is one mitochondrial mutation that mutant load has been really well characterized for. We don't have this type of data for a lot of the other ones, but it might be nice when we're looking at spectrum of disease over time. The mitochondrial deletion syndrome, so chronic progressive external ophthalmoplegia, Pearson's, and Kern-Sayers. So we talked about this. So the classic diagnostic criteria, which were identified actually by an ophthalmologist and a pathologist, not by a neurologist or a genetics person, but 1959. So an ophthalmologist recognized that he had patients around age of 20 developed paralysis of their eye movements. So you couldn't see it, and I didn't really do it with Liz, but that's part of the rest of her syndrome, and what we were looking at in clinic yesterday. So she actually has problems looking up, looking down, and looking sideways, and that is actually getting worse over time. She has retinitis pigmentosa, and then one or more of the following things were noticed: some type of cardiac arrhythmia, sometimes a gait disturbance, and some type of neuromuscular degeneration. And this is the classic criteria. Now, over time, what has also been associated with Kern-Sayers is endocrine and kidney problems. We know about the deletion. I talked to you about this already, but the syndrome can also be associated with other deletions, duplications, and it can be autosomal dominant, autosomal recessive, nuclear DNA mutations can also cause Kern-Sayers, although the most common thing is that it's sporadic. It just shows up. And we know that is in Liz's case because you would expect a mitochondrial DNA problem to come from mom, right? We tested mom, and mom doesn't have it. So if mom doesn't have it, assuming that her eggs are normal, because remember, we can't test a woman's eggs and we're born with everything we're going to have, but somatically in her body cells, mom does not have this deletion. Liz has it. Liz has 2 healthy brothers that don't have it. Okay, so these are some old pictures of Liz of her really droopy eyes close up and her inability to really look up. So one of the things when we first saw Liz was in order to have her to look up, she had adapted to doing this. So you would ask her to look up, and instead of being able to just move her eyes up, she would just move her whole head up. So her eyes went up, right? But really, if you hold her head still, now she can't even get them up this much. Now we're not really seeing white at all. And this is you before you had your eyelid surgery, so you didn't even talk about that. So she's actually had eye lift surgery because her ptosis got so bad that she was unable to read for school. Retinitis pigmentosa is seen as you lose pigment, so this is called a characteristic bullseye, so I put a circle around it. Dr. Zen in the Eye Institute took these pictures for us, and this is her optic nerve, and you can tell it's smudging at the edges, so that's consistent with the atrophy. All right, now to make life harder, there has to be crosstalk between the nuclear and the mitochondrial genome, which I had put up before, and this is the cause for, we think, a lot of the depletion syndromes, plus minus deletions. So there's actually a reduction in the mitochondrial DNA copy number, which I've sort of already mentioned. This group of genes is really growing, and this also can be dominant, recessive, or sporadic. So we sort of have already talked about this. All right, so here's the clinical dilemma. So the clinical dilemma is underlined as in potential. So we have significant limitations in diagnosing these disorders, because what we all learn early on is to look for lactic acidosis. But lactic acidosis occurs only in about 1/3 of the mitochondrial disease patients, and most frequently in the really severe younger patients, but not in adult patients. And so we have to look for other things. So is there anemia, meaning not enough red cells, or pancytopenia, meaning not enough of every type of cell, not enough white cells, platelets, and red cells? Are there some elevated liver enzymes, as in MPV17, you'll have severe liver disease. We can see that in a couple of the other mitochondrial disease patients. But these are not specific things. I think you can tell already, because you might have anemia because you don't eat well or whatever. We have low blood sugar. In mitochondrial disease, it tends to be what we call postprandial, meaning after a meal. Elevated CK, we talked about, uric acid. So all of these things we see sometimes, but not all the time. And so we have very bad biomarkers, really, in the things that we can get to easily, like blood and urine. Liz does not have lactic acid in her blood, but we've actually looked at her cerebral spinal fluid, and her lactic acid in her spinal fluid runs very high. And that's probably why she has stroke-like episodes and migraine headaches and some other things that we're trying to manage. There are specific enzyme problems. So if you do enzyme, if you test skin, you may or may not find an enzyme deficiency. If you test the tissue that has the most disease, so like liver or muscle, sometimes you'll get a better indicator of mitochondrial disease. But heart biopsies are not easy to do, and you do sometimes have heart tissue you want to look at. And we don't usually go and do brain biopsies on patients either to diagnose a mitochondrial disease. So if those are the tissues that have the most symptoms, you have a limitation. The mitochondrial depletion syndrome, or mitochondrial depletion and muscle CoQ content, can be used as potential biomarkers, but only a few places around the country do those, and that's not seen in every type of mitochondrial disease, and it can be seen in other types of muscle diseases, so again, not super specific. And then molecular confirmation, right now we know there's 2 genomes, so you have to look at mitochondrial DNA and nuclear DNA, and that's difficult. And so I talked about limitations, and here's by the numbers. And treatment, we've sort of already talked about with Liz. And there's lots of drug-induced mitochondrial toxicities. There's a whole book you can get available online for those things to avoid. And then basically there's Mitochondrial Cocktail, which used to include a lot of things, but now pretty much ubiquinol, which is the water-soluble form of CoQ, and carnitine, creatine, and alpha-lipoic acid are the only things that are really well studied. Tarnoplowski has done 2 really good studies, only with adults with electron transport chain defects in 2007 and 10. And then I just did a big review of the state of mitochondrial supplement research, if anybody wants to look at that. And I think we should hold questions and let Jennifer talk. So in summary, for any of you that want to do some research, I think there's some definitions that need to be clarified. We have very few natural history studies. We have difficult diagnostic strategies because we don't have good biomarkers and other diagnostic tests. And we have treatments that are really heavily focused on oxidative stress management, but not a lot of other things, and certainly no cures for any of these defects. And that would be Cassidy. That's my baby.
Thank you very much, Lynn. We have time for some questions, and perhaps if anyone wishes to ask Elizabeth one.
I think I just heard about gene therapy in utero for mitochondrial diseases.
Yeah, there's actually a number of gene therapy trials going on. There's actually one for retinitis pigmentosa in Florida. I think this is on. Can you all hear me still? Not really well? Yeah? Okay. So So there are a number of those, and I didn't put them on the slides because it's really pretty preliminary. But yeah, the whole prenatal diagnostic thing is a whole nother nightmare, and I took all those slides out. But if anybody wants to know about prenatal testing, I can discuss that at a different time.
Is the distribution of mitochondria from the mother to the daughter cells controlled, or is it by random?
It's completely random. That's the problem. Completely random. Unless you figure out a way to control it.
So what is the source of the disease for Liz?
So Liz has that mitochondrial deletion. So she has a chunk of her mitochondrial DNA in every cell of her body missing, lots of chunks out of lots of those DNA. And so we do understand her mechanism of disease well.
So how about some MRI of the brain or heart? Did you have a chance?
Her MRI of her brain does have some abnormalities that we would expect, some white matter changes, but chemically it was the bigger thing. So chemically, she has a high lactic acid in her spinal fluid, and she has cerebral folate deficiency, and both of those are treatable. So we've been giving her folinic acid and alpha lipoic acid to manage those, but that doesn't cure it, that just controls it. And her heart, her echocardiogram has been normal, but her EKG did develop heart block, so she has a heart pacemaker in now.
Is it biventricular or?
Yep, it is.
Thank you.
I deferred to the cardiologist who said biventricular is the only way to go these days.
So, Lynn, what happens when patients with mitochondrial disease have some acquired infectious process or immune process or some
Right.
other disease? How do they respond?
Right. So as you can imagine, an infection or trauma increases our body's need for energy, and they can't really mount energy on a normal basis, as you can tell by Liz. And so if you have inadequate energy to start with, and Mom didn't actually discuss this, but when Liz gets sick, she is sicker than her brothers would be or other kids her age, and she stays sicker for weeks. It takes her almost a month to recover from strep throat, right? So that is what we see typically for other types of mitochondrial disease as well. It really impacts on your activity, going to school, going to work. It's really hard.
You mentioned about the mutant load to wild type mitochondria. Do you know what is her load?
Could you repeat the question?
Yeah, so the question was, do we know the mutant load for Liz? And the answer is no. We don't know. So we did some heteroplasmy studies only in blood, and that was about 40%, but that doesn't tell us how much mutant load might be in her eye or in her muscle or in her heart, and so it's kind of hard to tell. We know there's tissue variability.
You mentioned ubiquinol as a complex 1 and 2 treatment. Is that just to make up for lack of normal ubiquinol, or is there some curative effect in there?
No, so it's really not curative, and it's actually not specific to complex 1 or 2. So that diagram, I sort of rushed through a little bit. So ubiquinol is the normal electron carrier between complex 1 and complex 3 and complex 2 and complex 3. So when you hear the electron transport chain is numbered 1, 2, 3, 4, 5, you think it goes in that order, but 1 and 2 really don't even communicate. So it basically goes 1 to 3 and 2 to 3, and ubiquinol is the electron carrier there. But ubiquinol in general, we use as a reactive oxygen species, MOP, basically, for lots of different types of mitochondrial diseases. So it's not curative and it's not really specific.
All right, I think we should move on. Maybe, do you want to make a last comment? Do you have anything you'd like to say?
Yeah, sure. It's kind of hard for me to really keep up with my friends because they have more energy than me, and I don't have enough to keep up with them.
All right. So we want to thank you very, very much for being here, both of you, today. Right. Okay, so Jennifer is the head of the chief of the section on organelle biology in an ICHD. Sorry, please.
That's me. Yeah, no, it is very sobering to talk after Lynn to see the impact that problems with this organelle that we call the mitochondria can give rise to. Now, what I want to do in the next, and I'll try to do this in 30 minutes, is to give you a perspective on the what we know in terms of the cell biology of mitochondria. And the hope is that by getting greater insight into the dynamics of this organelle and how it's behaving under different conditions of stress or starvation, that we might be able to get a better handle in terms of what's going on when you have these diseases like what Liz had or has. Okay, so mitochondria. Now, mitochondria are unusual, or I would say remarkable organelles within cells because they really represent an endosymbiont. And I guess if we get these lights down, it's going to really help you see some of these movies and everything. So this is a lifestyle movie of a cell that's been transfected with a mitochondrial fluorescent protein that targets to mitochondria. And you can see that the cell is just chock full of these organelles that we call mitochondria. The mitochondria look like they're moving around. In fact, they look like little bacteria that are swarming around within the cell. And the reason why they look like that is because that's in fact how they evolved, how they co-evolved with the eukaryotic system. So the idea is that mitochondria originated as a bacteria that was engulfed by an ancient archaic cell, and that together they acquired a host of new characteristics that essentially created what we call the eukaryotic kingdom. And what we think is that mitochondria are really in the same line as these proteobacteria that really trace an origin that's over 2 billion years separated from this eukaryotic cell line. And so a lot of people, a lot of evolutionary biologists now believe that what really defines the eukaryotic cell as a cell is this endosymbiotic event where a probacteria that is now known as mitochondria was engulfed by an ancient archaic cell, and together they acquired, they both essentially created a symbiotic relationship that allows the complex functions within the cell. So what the take-home lesson from that concept is that mitochondrial functions must be deeply integrated with the activities of the cytoplasm and the nucleus of the eukaryotic cells. Otherwise, these little protobacteria would have been, at some point, sort of lost from cells. But every single eukaryotic cell that exists on this planet has mitochondria or mitochondrial genes associated with them. So this organelle is deeply integrated in terms of its activity with the host, with its host. So what are some of that integration? Well, you heard the list from Lynn of all the things, a subset of all the things that mitochondria do, which includes iron-sulfur cluster formation, apoptosis, calcium regulation, heme biogenesis, cholesterol metabolism. But this chart right here gives you a flavor of the cell metabolism that mitochondria are participating in. So everybody thinks about this oxidative phosphorylation or electron transport chain where mitochondria can produce in a very efficient way ATP. But in fact, when cells are exposed to glucose, lots of glucose, and are not starved or stressed in any condition, the carbon chains that come into that mitochondria and move through this TCA cycle are, by and large, being used in anabolic processes to build, for instance, lipids or amino acids or nucleotides. So a lot of the energy from glucose and sugars that come into the cell are being used by mitochondria to actually create biosynthetic components of the cell, lipids, amino acids, and nucleotides. It's only under special conditions that I'm going to be showing in this talk where cells really start shifting to rely fundamentally on this oxidative phosphorylation function of this TCA cycle. So mitochondria are really, you can understand how they are fundamentally embedded in terms of all of the biosynthetic and metabolic pathways within the cell. Now what's intriguing about mitochondria is that they themselves can undergo a fission-fusion cycle. They don't just sit around as 200 little sort of bacteria-like structures that are moving around the cell, as I showed in that movie. They are capable of actually fusing as well as undergoing fission. And this is regulated by genes that drive fusion, and these include mitofusin 1, 2, and OPA1, and gene products that drive fission. This is a dynamin-related protein, DRP1, and I'll be talking about these proteins that are regulating this fission-fusion state. Now, one of the things that we know is that when mitochondria are highly fused, they are more likely to release cytochrome C and initiate the apoptotic response that leads to cell death. When mitochondria are more highly fused, cells seem to be protected from this process of cytochrome C release and apoptotic stimuli. So the cell is clearly interested in regulating when to have cells, mitochondria fused versus fragmented. Well, a postdoc in my lab by the name of Kasturi Mitra was intrigued enough to address the question of, well, what's the state of mitochondria at different stages of the cell cycle? Do mitochondria change their shape in any particular way? And is this important for the cell to go through its cell cycle? Well, what do I mean by cell cycle? Cells essentially have two really separate states. They have a state where they're essentially dividing, separating, and that's mitosis. And they have a state where they're duplicating their nuclear genome. And this is called S phase. And these intermediate stages, which are called G1 and G2, are gap phases where the cell is neither dividing nor duplicating its DNA. It's essentially just hanging out, so to speak.
What is the cell lineage?
Pardon?
What is the cell line?
This particular cell line is an NRK, a normal rat kidney cell line, but we've done this in a variety of different cell lines. The upshot from what Kasturi found is that mitochondria, which are shown in red here, in each of these stages of the cell cycle, undergo a dramatic change in their morphology. Specifically, during mitosis, mitochondria fragment into small individual units, about 200 to 300 units, and that we believe is important for the cell to be able to partition mitochondria efficiently as the cell divides in two. At the other extreme, this S phase, mitochondria become highly tubulated and interconnected. And in fact, we've shown that they essentially fuse into almost a single system. They become hyperfused into one interconnected network. And in G1 and G2, they essentially are intermediate between this highly fused state, which occurs right at this G1S boundary, and the highly fragmented state in mitosis. And just to illustrate how dramatically this change is in terms of the mitochondrial morphology, here's a cell showing mitochondria in red that is in G1S. And you can see that the mitochondria have essentially fused almost entirely together. 90% of the mitochondrial mass is just a single structure. So there is clearly a dramatic regulatory change in terms of the fission-fusion dynamics as a cell is preparing itself to replicate its nuclear genome in S phase. Well, Kasturi then went on to determine, well, is there anything special about this highly tubulated mitochondria? And that she observed at G1S. And remarkably, what she found was not only was the outer membranes of the mitochondria fused at this stage of the cell cycle, but the matrix, which is this internal region here that's surrounded by this inner membrane, is also continuous throughout this whole fused network. There's increased membrane potential associated with these inner membranes, suggesting that the electron transport chain is more active, it's producing more ATP, and consistent with that, we can absolutely see increased ATP generating capacity of these organelles. So what this suggests is that mitochondria are undergoing a cell cycle dependent change in their morphology that is somehow boosting ATP and membrane potential of these organelles right at this critical stage of G1S. So the question immediately arises, well, is this change in mitochondrial characteristics in some way important for the cell to actually go through S phase? And so to test that, what we did was we treated cells with a drug, it's an ionophore, FCCP, that causes mitochondria to fragment. And when we treat the cells with FCCP, what we find is that cells cannot go into S phase. What we're looking at here is an S phase marker, PCNA, which labels the DNA replicative foci in the nucleus at S phase. And you can see normal untreated cells, if you start imaging them in late G1, within 4 hours, they're going into S phase, and you can see that with this PCNA marker. By contrast, if we do parallel treat the cells with FCCP, you can see that they never go into this S phase. So 10 hours later, the nucleus is still showing no signs of any type of replication. And this is just a different type of experiment where you're looking at BRDU incorporation into the DNA to determine DNA replication. And you can see if we add FCCP to cells in G0, we do not get BRDU incorporation, in contrast to control cells within about 3 or 4 hours after incubation, starting in G0. So what this is suggesting is that the transition of the mitochondria from this highly fragmented, this dynamic tubulated fragmented state into this predominantly tubulated state at G1S is in some way critical for cells to go into S phase. It's almost as if it's a type of a checkpoint. And consistent with that, we see that P53 is absolutely required for this. You need increased P53 in order to get this transition. We can also, we see a correlation with cyclin E, which is the S phase cyclin during this process. So if we can actually initiate a G1S checkpoint by essentially interfering with this process. Okay, well, if this highly fused state is important for cells to move through S phase, can we induce cells to go into S phase simply by manipulating the mitochondrial morphology? And so one way to do that is to interfere with the fission machinery, to interfere with this dynamin-related protein that is known to be responsible for driving highly fused mitochondria into this fragmented form. And you can do that by either expressing a mutant form of DRP1, overexpressing it, or by adding a drug that interferes with the activity of DRP1, called MDV1. And so what we decide, if we added the MDV1 in cells that essentially these are cells that are in G1, and we add MDV1, what you see is within 4 hours, we start accumulating cyclin E, which is the cyclin that is responsible for S-phase progression. And as you can see, the mitochondria become highly fused. So this suggested that we're actually pushing the cells into the cell cycle, into S-phase, by causing mitochondria to become highly fused. So in order to more rigorously test this, what we did was we took cells that had been pushed out of the cell cycle by serum starvation and into this phase that's called G0. They're no longer cycling, these cells, because they don't have serum. And we asked, could we take these cells out of G0 and drive them back into the cell cycle by manipulating mitochondrial morphology, by making mitochondria more highly tubulated? And so we did that by adding this drug, MDB1, to cells that are in G0 through serum starvation, and then looked for BRDU incorporation, which indicates replicating DNA. And here is the result of this experiment. This panel shows DNA in blue and BRDU staining in red. In just G0 serum-starved cells, you can see none of the nuclei are essentially replicating their genome. None of them are in S phase. Whereas, by contrast, after 3 hours of adding this drug, which causes mitochondria to become highly fused, you now see that a large fraction of these nuclei in these cells are replicating their DNA, indicating that simply by causing mitochondria, driving mitochondria to becoming highly fused, can force these cells into S phase, can drive them through the cell cycle. So this was pretty exciting when we made these observations. I mean, we were really excited because we started thinking that maybe this is somehow connected to the decision by cells to proliferate or differentiate. Maybe mitochondria are playing a role here. So the idea is sort of diagrammed here. I mean, cells proliferate, and then at some point in the life cycle of these proliferating cells, they decide to stop dividing and differentiate. At that point, they go into G0. And so what we wanted to know was whether we could impact that decision of cells to differentiate versus continue to proliferate during development by manipulating mitochondrial structure, mitochondrial fission-fusion state. And so to test this, we used the fly oocyte as a model system, because it's very easy to knock into these follicle cells that are essentially an epithelial cell layer that surrounds the oocyte and these nurse cells. And we can knock in null mutations of DRP1, which means these cells will not have fission machinery and mitochondria will become highly fused. Or alternatively, we can silence a mitofusin, which is responsible for fusing mitochondria, and the result is that mitochondria will become fragmented. And so we're going to ask, if we introduce these mutants in these follicle cells here, can we alter the fate of these follicle cells during development? And the answer is yes. It's incredibly exciting. Here is this is a normal egg chamber where you can see these follicle cells are just a single epithelial layer of these follicle cells. Blue labels the nuclei. If we express a null mutation of this DRP1 so that the mitochondria now are going to be highly fused together because you don't have fission going on, what you now see is that the cells proliferate dramatically. And so you just have a huge mass of cells that are continually dividing under these conditions. You also, if we knock it into areas of the egg chamber where the cells should be differentiating into like these stalk cells here, you lose the ability to make these stalk cells because the cells are proliferating. They're not dividing any longer. And all we've done is just changed the morphology of mitochondria so that they're more highly fused. Now we can do the opposite experiment. We can knock down a mitofusin, making the mitochondria highly fragmented. And under these conditions, if we look at a differentiation marker, we can see that mitochondria are actually differentiating earlier, prematurely differentiating under these conditions. So what we've been able to illustrate in this follicle cell model system in the fly is that by manipulating mitochondrial morphology, driving the mitochondria into a fragmented or fused state, we can shift the cell fate of that epithelial layer to one where the cells are prematurely differentiating or one where the cells are proliferating much longer than they should. So this suggests a really important role of mitochondria in cell fate determination, and we think that this is potentially playing itself out during development in a big way in a lot of different tissues. In this, if you're interested in this, this paper has a lot more packed into it in terms of how how the fragmented mitochondria actually talk to notch signaling pathways and hippo. I don't have time to go into that, but we think that at the heart of these control pathways is the physiological state of the mitochondria. Okay, so as I mentioned, we think mitochondrial state regulates cell cycle progression and cell fate determination. Well, do other cellular states require highly fused mitochondria, or can they be impacted, or do they need highly fused mitochondria? And for this, Kelly Rambold in my lab made the observation that when she starved cells, she found that mitochondria became highly tubulated and fused together. So in an untreated cell in G1 or G2, the mitochondria are constantly moving around undergoing a fission and fusion. But when you starve these cells, and we're starving, we don't have serum and we have very low glucose, the mitochondria now fuse into this very highly integrated network. And so that led us to start thinking, well, maybe there's something going on with this network that is helping the cell survive starvation. So let me just emphasize what's happening under nutrient deprivation. The data from several labs is now suggesting that the fission machinery is actually being inactivated under starvation conditions. And so as a result, the mitochondria become much more highly fused together, and you get this phenotype we're talking about. Now, what does this phenotype do? And I'm going to shift to yeast, where Arnold Co. in the lab has been doing some pretty exciting stuff looking at glucose starvation and what it does to mitochondria in yeast. And so if you shift yeast from a 2% glucose diet to a 0.4% glucose diet, what you see is that the mitochondria actually become much large. The mass of mitochondria increases dramatically, and the mitochondria become highly fused. So again, starvation is causing mitochondria to actually grow, increase their mass, increase their highly fused characteristic. And this is just showing that you can see increased transcription of mitochondrial fusion genes under low glucose in yeast. So what's going on here? Why is it, what's the tubulated mitochondria doing that's good for the cell under starvation conditions? What we think is happening is that when cells get starved, that triggers AMP kinase activity, which inhibits mTOR, which is controlling growth, and then starts interfering with this mitochondrial fission-fusion machinery, and ultimately you get mitochondria becoming highly tubulated. Well, what does that high tubulation do? Well, one thing it does is it seems to boost cellular respiration, and that's illustrated here, where with 0.4% glucose you can see increased oxygen flux per unit cell. So having a highly tubulated mitochondrial system seems to really boost your ability to use oxygen, flux oxygen. And you need this mitochondrial fusion to survive low glucose. So if we prevent the mitochondria from becoming highly tubulated under starvation conditions, you can see the cells die. And this is just, we're inhibiting with these mutants that block mitochondrial fusion. And you can see that the cells die very quickly under 0.4% glucose, unlike normal cells. Well, if you look at what's going on under these conditions with glucose starvation, you can see that the mitochondria, these are in red, are highly tubulated. And if we look at an autophagic marker, LC3, you can see that the mitochondria are essentially protected from mitophagy. They're protected from being destroyed by the cell's self-eating mechanism called autophagy. And this is in contrast to cells where we've interfered with mitochondrial fusion by knocking it down. MFN2 has been knocked down. Mitochondria are now fragmented. You can see they're red, these red little fragments of mitochondria. And all of these mitochondria are now being eaten by autophagosomes in a process called mitophagy. And in fact, you can pretty much wipe out your mitochondria under these conditions by essentially preventing mitochondria from becoming tubulated under starvation conditions. So the tubulated mitochondria under starvation conditions, which is driven by this cell energy sensor system, is not only boosting ATP, but is preventing mitophagy. So it's allowing the mitochondria to actually survive. And we've also found that, remarkably, the tubulated mitochondria is driving non-selective autophagy. And we know this because we've been looking at autophagy. This is a autophagic marker, LC3, which when you look at it as cells are starved, you can see that there's a dramatic increase in your total autophagic vacuoles in cells under starvation conditions. And when you look at where these autophagosomes come from, they come off the surface of mitochondria. So to the left is mitotracker, which is a mitochondrial marker in red here. And in green is LC3, an autophagic marker. And you can see that as the autophagosome forms, it's forming off the surface of these the mitochondria. So what this is suggesting is that the tubulated mitochondria is really doing three things under starvation. It's because the mitochondria become electrically coupled, it can produce ATP more efficiently, and so you shift to oxidative phosphorylation as your major energy supplier. But at the same time, you're protected from mitophagy. You're also protected from apoptosis. As I mentioned, tubulated mitochondria do not release cytochrome C very efficiently. And finally, the tubulated mitochondria, we believe, drive a non-selected autophagic process, which ultimately releases free amino acids and fatty acids and other components that allows the cell to actually start rebuilding itself under starvation conditions. And ultimately, under these acute starvation conditions, we can see the cell adapt. So you can get a feedback mechanism here where you actually slow, the mitochondria are essentially driving the show under these starvation conditions, and the cell's pretty happy for quite a long time. Well, how can the cell be happy for quite a long time when it's being starved? Well, glucose and glycolysis are shut down under the conditions of starvation that I'm talking about. But the cell is loaded with lipid droplets. These are fat globules that have accumulated during glycolysis. And what's going on under starvation is that the cell is shifting its metabolism so that it can now start breaking down these lipid droplets, taking these fatty acids, feeding them into the mitochondria, and then driving the TCA cycle so that you create this much more efficient ATP release or production from mitochondria. So the tubulated mitochondria, we believe that are induced under starvation conditions, are playing a multi-pronged role in cell survival, in addition to driving autophagy, which is critical for the cell to actually reboost itself, to start making things it really needs under starvation. It's protecting the cell from apoptosis as well as mitophagy, and drives an efficient oxidative respiratory mechanism. And so with that, I want to just end by thanking the people in my lab who've been really driving all of this work. I want to thank Kasturi Mitra, who really initiated all the mitochondrial work in my lab. She now has a great job at the University of Alabama. And the people in my lab that are carrying on that work include Kelly Rambold, who did the work in the mammalian cells, initially observing the tubulated mitochondria under starvation, and Arnold Seo, who's doing the work in yeast. I also want to thank Sarah Cohen, who's been looking at the lipid droplet connection, and Dale Haley, who's done all of the work related to mitochondria and autophagy that I talked about. We collaborate with a variety of different people, the University of Florida, and at NIH, Wynn Arias, we actually have some really interesting data related to the role of mitochondria and cell polarity, the generation of cell polarity. So with that, I'll end and thank you for your attention, and we can have some questions if you want.
Thank you very much, Jennifer. Jeff, can you put the light? Oh, thank you. Yeah, go ahead.
Yeah.
So, is there any protein that helps this under starvation condition mitochondria to fuse, or they are doing it on their own? So obviously there must be a structural changes that is happening.
Oh, absolutely.
Is it induced by certain proteins, or it is just structural changes in each mitochondria?
So we think that it's a combination of both the fusion machinery being activated, but also inhibition of the fission machinery. So the machinery that normally causes mitochondria to break apart, we think is being inhibited. And so the cells, the mitochondria don't break apart, and they're more efficiently fusing. And so you get this shift, this entire shift of the mitochondria from this sort of dynamic fission-fusion organelle to one that's more predominantly fused.
So as I was looking at the influx, you have the glucose and the glutamine, they are getting in. So you think if they are used as a nutrient, it also could be used in the patients with mitochondrial disease to get rapid nutrition?
Yeah, so that's a really cool question. I was thinking about that myself while I was hearing about all of this. So in terms of Liz, you know, what's going on with Liz? I don't really know. I mean, this is the first time I've heard anything about her syndrome. But it sounds like she can't, I mean, she cannot do this oxidative phosphorylation. She's got a real problem in utilizing this oxphos. Well, she's doing glycolysis. She's surviving off of glycolysis. So she's being fed. I mean, that's why she's got to be on her GI tract. She's living off of just food that's essentially constantly driving the glycolytic pathway. And actually, I should just go back to the first slide, sort of showing. I think that's the easiest way to look at all of this. Here.
Oh, you got it?
I've got it, yeah.
Yeah.
So, so as long as she's got sugars that she's being fed, and and proteins, they're driving this glycolytic pathway that can push the TCA cycle at least to the extent where you get citrate to make lipids, amino acids. Not clear how effective, she's clearly not getting effective oxidative phosphorylation.
So the best thing should be to give her high glucose rather than actually.
Actually not, because in any of the mitochondrial disease patients that have lactic acidosis, if much sugar into the system, you actually make their lactic
Yeah.
acidosis worse.
Yeah.
So that's why we actually are
Just go into
giving her
these-
a lot more protein.
Yeah. Okay, so that's really good, because if you get too much glucose in here, she's just going like this.
Right.
So you really will just fill in these components that the mitochondria normally would be making that she's not making very well.
Right. So the interesting thing about this work is obviously, and I don't know what Danita was thinking, so one of the therapies that I didn't go into at length is on exercise.
Can you speak?
Oh, so in the treatment of mitochondrial diseases, one of the really exciting research areas is the use of exercise. And the reason that we promote endurance training is that it promotes wild-type biogenesis. So I'm wondering whether or not so much from the perspective of stopping starvation as a management for patients, but whether or not pushing the fusion and cell replication would increase their mitochondrial biogenesis
Exactly.
at all.
Right.
And that may actually be a great cure for a number of the mitochondrial diseases, because some of the newer drugs and exercise therapy and some of the other things that we're now looking at for treatments really are pushing and focused on mitochondrial biogenesis.
Have you tried fasting at all?
Yeah, so calorie restriction has long been utilized. The problem is that in a healthy individual, calorie starvation and in mice and rats promotes longevity.
Yeah.
But if we starve Liz,
No, she can't.
yeah. So in our truly affected mitochondrial disease patients,
Yeah.
starvation does not
But
help.
with Liz, too, I don't think exercise is going to do the... She's in a different category, it seems to me.
She is, because she took out 3 complexes, she's
Yeah.
severely impacted. Yeah.
So, Jennifer, what happens to mitochondrial DNA during fusion?
That's a good question. We don't really know. I mean, people are trying to look at that. It's probably more interesting what happens during fission, when mitochondria
Yeah.
break apart. And right now, there's a lot of people who are looking at this. I think there's going to be some papers coming out. But the thinking is that the site where the division is occurring is a site where the DNA is actually replicating or has replicated. So that, because every fragment of mitochondria seems to has DNA associated with it. So there's a very good coupling between when a mitochondria breaks into 2 and the DNA goes with it.
But if the proportion of normal and abnormal
There is probably mixing and,
Yeah. I'm sorry. If the proportion of normal and abnormal mitochondria is stochastic, what happened? It's conceivable that what happens during fission and fusion, does that representation still represent whatever it is, 50% or 80 or 20%?
No, yeah.
Or is there perhaps
Well,
some
when
way
they
of
fuse,
facilitating
you now have
segregation?
lots of DNA associated with it when they fuse together. And the issue is there some way that you could, can the cell recognize mitochondria that have some sort of perturbed, damaged DNA and selectively get rid of it by mitophagy? Yeah, so that, I mean, that would be an interesting possibility, but so far, nobody's clear on that.
Jennifer?
Yes.
Yeah, I wonder if you can reconcile the concept that, if I understand correctly, if you induce artificially fusion and you have this long tubular mitochondria, the cell is driven to S phase and division. Yet under starvation, you're basically getting the same tubular formation, and of course, the cell goes into G0. So how you reconcile that?
Because,
what do you think?
In one situation, the cell is well fed. When a cell is proliferating, the cell is under a well-fed condition, and there's serum. Under the conditions of starvation, there's no serum and there's low glucose. And so we're seeing a similar change in mitochondrial morphology in each case, at least at the level that I've talked to you about. Now, exactly whether they're identical in terms of whether the electron transport complexes are in a super complex or how they're arranged, we don't know. We have to get at another level of magnification, so to speak, to understand that. But clearly, they're both, the highly tubulated state has a different function in a cell that's starved versus a cell that's well fed and under serum, driven under serum.
And you can't detect any differences between those two states in terms of...
We haven't, I think we haven't really looked hard enough. We should definitely do some more experiments like photobleaching and looking at diffusion mobility, how connected the matrix is.
How is the mitochondria gene distributed in that body? How is it distributed?
In here?
Yeah.
It's somewhere in this matrix. So there's been a this past year, there's been a super resolution imaging paper from Harold Hess where they've looked at matrix markers and nucleoids. And they don't see anything particularly unusual. You just see a big blob of DNA sitting in here, sitting somewhere in. So far, we haven't been able to relate it to anything in particular. Yeah.
I had a question concerning, you said that when the mitochondria fuse, the membrane potential goes up.
Yeah.
So I was wondering, have you actually measured that? Because if it goes much above 200 millivolts
Yeah.
or some, your mitochondria become leaking, and actually your production of ATP becomes less efficient.
Exactly.
And a follow-up to that, do you know, for example, what happens to the cristae? Because when you fuse, where you actually probably have the ATP synthase complex, kind of what maybe
Yeah.
you can comment on that.
So Kasturi did these measurements using TRME, and she did a ratio analysis of TRME to mitotracker green to look at the increase at S phase relative to the different other phases. And it goes up about 2 or 3 fold. But that's really different than if she hyperpolarizes the membrane where it goes up like 10 or 20 fold. So we don't think it's causing any kind of leakage, well, the kind of leakage that you would get under a hyperpolarization scenario. But definitely, we have firm quantitative data that the membrane potential is going up by at least 2 or 3 fold.
By taking IPSL, you could have a lot of fun from Liz.
We do have an IPS cell. Yeah.
Would the ADP will be held for the patient? It would give the ADP to the patient with the help?
Just give ATP to a patient?
Mm-hmm.
I don't think so.
Yeah, there's not really, it doesn't work like that, yeah. The closest that we get, which we really didn't discuss too much, is we do supplement with creatine. So after illness, when Liz is having a really hard time recovering, we can give creatine monohydrate powder, which actually all the muscle builders use. But in this case, we use it as a storage molecule for energy, for creatine, broken down by creatine kinase to try and boost her ATP. How efficient that really is cellularly, I'm not really sure, but theoretically it should work, and it seems to make a difference when she's really, really exhausted.
Second question is how to count the mitochondria? Is counted is number or counted function? Functionally, you count the how many percent mitochondria to do the business.
Yeah.
How do they count?
Heteroplasmy, you mean?
Yeah, how to count the number?
So her heteroplasmy in her blood was measured just looking at how many of the mitochondria, the wild-type mitochondria as percent compared to the percent of mitochondria that had the deletion. So, and it was only in her blood, and in blood, it's basically what the blood cells. So it wasn't measured in any other tissue, and so we have a hard way of really making any meaning out of that.
So how to count it in other tissue?
So you have to take a sample, you can do it. You have to take a sample of all the other tissues and actually ask the same test to be done. So they do something like PCR or something to get a handle on that. Believe it or not, the 2 tissues that give you the most accurate is bladder wall cells and bulbs from hair. So I didn't pull any of her hair out. And we tried a few times just to see if we could get bladder cells with a regular void, and we just didn't get enough. So sometimes you go in and get irrigated recipe, and we just not going to go there.
Maybe I misunderstood, but for your fusion data in relation to starvation, you weren't showing that in case of starvation you get more fusion. Am I correct?
You definitely get more fusion. That's the only way that you get more. That's the only way you can get more tubulated.
But I thought you were also interfering with the fusion,
Yeah.
and you were showing fused ones are protected against mitosis.
No, that's a good point. So one possibility is that all that's happening is you're blocking fission.
Yes.
But we've also seen using fusion mutants that fusion genes are important as well to make this process efficient.
But have you shown that in case of a starvation under electron microscopy, you get more fused mitochondria, or any other method? Have you shown with only the starvation, no other manipulation, you get more fused mitochondria?
Yes. They're more, the mitochondria shift from being fragmented to being much more highly fused, and we can quantify that under starvation conditions, definitely.
So can I ask a quick question while I'm walking down here to get it? So if the problem in the mitochondrial diseases is basically a reduction in mitochondrial number or function, rather than something that the, quote, bad mitochondria are doing to inhibit the good mitochondria, I mean, it's a depletion thing. So is there any role then for possibly enhancing mitochondrial biogenesis through things like the sirtuin mechanism and PGC1 alpha and FOXO that actually drive mitochondrial biogenesis? So you have mice and all with knockdowns of these models. Has that approach been considered?
Yes, and interestingly enough, those are exactly the chemicals that are upregulated in starvation. So it's sort of a catch-22, but yes, those things are actually being studied quite a lot. We've actually seen a fair number of presentations at the mitochondrial disease meetings the last few years on C2E1, but it's not ready for patient delivery yet.
So I was curious, with your really interesting data on the role of the mitochondria in cell cycle progression, we already know they play such an important role in metabolism. Given that, I was curious about whether, and Lynn had mentioned that their DNA repair mechanisms are not as sophisticated as in genomic DNA, whether accumulation of mitochondrial mutations plays a role in carcinogenesis?
I would probably think so. I mean, absolutely.
Yeah, if you look at the cancer literature, that's actually pretty well described. So there's certain cancers that have depletion, but there's also certain cancers that are actually associated with certain mutations in the mitochondrial DNA. And the cancer literature has really covered that well.
Well, I think the perfect example is P53.
Yeah.
So P53, in addition to its role in cell cycle control, is also regulating a lot of mitochondrial genes. And so we think that that's very much connected. And we think that actually cells go one way that cells are sort of making a decision, do we have enough energy to actually go through S phase so that we can efficiently replicate the genome. And the cell makes that decision based on a number of things. Does it have enough nutrients? What's its metabolic state, et cetera. And we think that mitochondria is key for that. So if the cell doesn't have enough energy, the cell goes into a G1S block. And P53 is critical for that. If you knock out P53, the cells just go through the cell cycle. And I should emphasize that when we knocked down P53, we were unable to get to see the mitochondrial-induced cell cycle block. Remember I said that we could fragment mitochondria and cells would not go through S phase? Well, They do go through S phase if we don't have P53.
So it seems to be downstream of the fusion.
Exactly. They're definitely talking to each other. Yeah.
I had a quick question about your model in the fly. So you induce proliferation by inhibiting the fission machinery, but in the normal cell cycle you showed that fission was necessary for cell division. So I was wondering if you understood how that worked.
Okay, yeah. So we're seeing this particular phenotype where we're inducing proliferation by causing the mitochondria to become fragmented. In this particular area of the follicle cells where those cells are receiving growth factor stimuli from the oocyte. So the oocyte releases gherkin, which is an EGF-like growth factor, and that is what actually triggers mitochondria to actually undergo fragmentation, and that is a trigger to differentiate those cells normally. And if we interfere with that, the cells don't differentiate. They just keep proliferating.
Okay, well, I want to thank both of you very, very much on behalf of all of us. Thank you very much. That was
thank you.
very good.